Hi, everybody. Thank you for joining us. Dr. Joseph Barascano. I'm talking on behalf of IGeneX tonight to kind of give you an update of what's the year was like in 25 and what to look forward to in 26. Uh, you know, 25 was a record year in terms of number of tests that IGeneX performed and number of positives. So we see that tick-borne diseases from that point of view keep increasing. In Maine, Lyme, Babesia, Anaplasma hit record levels in 25. The climate and landscape changes are driving more tick spread and tick-borne diseases. There's been a first reported death from alpha-gal red meat allergy. And the CDC estimates are still probably not very accurate because they go by old-fashioned surveillance criteria, and we think that the number of true cases are even higher. But the point is that tick-borne diseases have not gone away. In fact, they're getting worse. In fact, this slide shows you that the surprise to me, IGeneX in the past 18 months have gotten samples from 36 different countries. And also they're finding the so-called European species like Arenii and Afzelii in positive United States samples, in fact, from patients who've not traveled overseas. So, to say that Lyme is a single illness from one germ in one part of the world is wrong, and this is proof of that. A lot of advances were seen industry-wide. One big thing, of course, is artificial intelligence. That's being experimented with to see if it can improve test reliability. I know IGeneX is at least looking at this now. The House and Senate Appropriations Committee approved increases in funding tied to Lyme. IGeneX immunoblots, both for Lyme and tick-borne relapsing fever, also Bartonella and Babesia, are now available worldwide and in all 50 states. In fact, the Lyme immunoblots, both IgM and IgG, are now FDA cleared. The at-home testing is now available through AccuDot, a branch of IGeneX, available in the United States and Canada. And this is simple. It's a home test kit that people can order online without a doctor's prescription. You take a blood sample with a finger stick and send it to IGeneX for processing. And the rapid Lyme test that IGeneX is developing, it's in a prototype stage. And what that'll allow is that clinicians can have a setup in their office where they can do a fairly accurate Lyme screening test right there in the office. Interestingly, I mean, shows how old I am. 2026 will be IGeneX's 35th year in business. Imagine that. I didn't think I was even that old. Now, a lot of you have seen this slide before. I bring it up because it's important. It shows about what we call co-infections or concurrent infections. In 2018, IGeneX compiled over 10,100 or so patient samples, and they found the percentages you see here. The most common was Babesia, with more than a third of the cases. About a third was positive for Lyme Borrelia. Surprisingly, almost 28% were positive for relapsing fever Borrelia, and you can see the rest. And what I think is even more important is that at least 40% showed positive for two pathogens, and as many as 5 were seen in some of these. Now, the note about this is IGeneX is a reference lab. So, of course, the sicker patients are represented here. So maybe this is not a statistic that applies, countrywide. But on the other hand, this data is from 2018 and before the more advanced recombinant assays that IGeneX has developed came online, and certainly before the culture and the improvements in the FISH test. So if this study were repeated now, I think the numbers would be even more impressive. What's available to test for Lyme? Well, I like to break them down to make it simple. It's what I call standard serologies of the old mainstay, the ELISA, the IFA, the Western blot, and even the microarrays, those well tests. I call them standard serologies. I don't recommend them because they're made up of protein antigens. It's how these tests are made, but they come from a single species, a single strain, and it's a lab strain, not a human strain. So it's really not very accurate, and I'll show you data on that in a second. As opposed to that are the advanced serologies that don't use bacterial antigens from cultures. They use laboratory-created recombinant protein antigens. And this allows them to be not only more accurate, but they're multi-species, and the antigens come from clinical specimens. So it's much more a real-world type of test. IGeneX offers these as what they call the Lyme screen assays, a screening test. That's tier 1 test. The broad coverage assay, which is an ELISA replacement. AccuDot, the home test, and the immunoblots, which are pretty famous at this point. They also offer the T cell testing, the T cell mitogen assay. A serology is a B cell based test. This is a T cell based test. Um, and it can be used in all stages of the disease. In fact, very early, I'll show you some information on that. The FISH test, the fluorescent in situ hybridization, is a very popular test. It is a test where the blood is put on a slide and you look at it directly to find the pathogen. In this case, they have it for Bartonella and Babesia. And they use not simple dye stains, but they use nucleic acid stains that are very, very highly specific. So that's a very popular test. The PCR is a nucleic acid detection. It's very specific, but insensitive. So what IGeneX has done is they've modified that by culturing the blood. It's the first lab that will culture the full range of tick-borne disease pathogens. That includes Lyme, tick-borne relapsing fever, Babesia, Bartonella, Anaplasma, Ehrlichia, and the Rickettsias. And once they have the culture growing for 2 weeks or so, any positive is noted and then it's confirmed with a separate PCR that's actually specially designed for this. So it's a very interesting test, and it's picked up a lot of strange pathogens that no one expected to see in poor patients. So let me tell a bit more. Why is the ELISA not good? So the way a test works is you have to take antigens from the pathogen, add the patient's serum, and if the patient had antibodies, then there's a positive reaction. So the ELISA and the IFA, where do these antigens come from? They come from a lab strain, a single species only of a strain of the organism that is grown in the lab. It's not a patient strain often. And how they make this is they'll usually take the whole organism, in the case of Lyme, the Borrelia, and they sonicate it. They break it up into millions of little pieces. And this soup that results is what's put in the test system. So now, of course, it's going to contain antigens that are specific to the germ you're looking for, let's say Borrelia or Bartonella. But it's also going to contain a lot of non-specific antigens, protein and otherwise, that might be found in other cells. And so what you get is a very non-specific test where you can pick up antibodies from a lot of other different infections, and even some autoantibodies in autoimmune diseases. So the problem is that you have this overly positive test, and what the labs have to do to make it more specific is they have to turn down the sensitivity to get rid of this background noise. And that's that trade-off between sensitivity and specificity. And that's also why the testing, when you get a test that's handled this way, you get very low sensitivity, really inappropriately low. To get around this, some labs decide to use just one antigen. You can do, for example, in a Lyme test, flagellin. In relapsing fever, you can use GLP, but it's just one antigen. It's trying to do a Western blot with one band. Not every germ, not every pathogen will make that, and some others will make that aren't specific. So it's really not a good test. And for all these reasons, we just don't recommend these old-fashioned tests. Now, the microarray, you've seen some labs use this. It's multi-well technology. It's basically a modification of the ELISA, so it's really not that much better. Again, they use lyso organisms. Sometimes they cherry pick which ones they're going to use. But the problem is it's really not any better than the ELISA, which it's based upon. And published studies have shown a sensitivity of 50 to 57%, which is comparable to the two-tier test, which is also not very good. How about the Western blot? That's supposed to be an advanced test. Well, it's old technology. Again, they sonicate a whole organism. And that releases all sorts of antigens. And what they try and do to clarify it, they'll separate them out by electrophoresis. So individual antigens are now spread out on a test strip. And you can see that in the picture on line 8, that's their positive control. So this represents the different antigens that have been released. So if the person's blood that's sent has antibodies, it'll bind to these antigens and make a dark spot. These are what we call the bands. So how do you interpret this? Well, you have to line them up. So lane 1 is a negative control, which is nice and negative. Lane 8 is a positive control, and that's your reference. So the other ones in between are patient samples. So like if you see these lines, where do you line these up? I mean, if I was a lab tech, I'd get more gray hair trying to figure that out. So this uncertainty can certainly impact accuracy. Also, when you break up an organism, as I said before, a lot of these non-specific antigens come out, and that's what you're seeing in this lane number 8. And some of them are specific for the organism, and some of them are not. They cross-react with everything else. And so that makes it very uncertain. And finally, How dark the spots are represent not only how much antibody is present in the serum, but also how good was the donor culture of organisms that were grown in the laboratory to make this thing in the first place. You have a poorly grown culture, you get weak bands. You get a strong culture, you get strong bands. So it's very nonspecific. And again, it's based on one species, one strain, usually a lab strain. And because of all these reasons, it's not very specific, and the sensitivity really could be better. So what IGeneX has done is, instead of using these sonicated organisms, they use what are called recombinant antigens. They're lab created, highly specific, and they're purified. And what do they do? It's kind of reverse engineering. Let's say you want to make recombinant antigens from Lyme disease, from Lyme Borrelia. So they look at the genetic sequence that the Borrelia have that creates these specific antigens, and they clone that, deliver it to E. coli with a viral vector, and then grow it in a very highly purified fashion. Then they go through many more purification steps. So you can actually pick and choose exactly which ones you want. And it doesn't have to be one, you can do a lot. And in fact, in the Lyme immunoblots, there are over 30 specific antigens that are put there to make it much more reactive in the real world. So what does this do? It means the test is much more specific to each organism. You're not getting this, background soup of random things. So you really almost completely eliminate false positives and false negatives. And because you use many different individual proteins, not only does that increase sensitivity, but they've been able to pick and choose proteins from a variety of species. So you're not doing one strain of one species of a lab strain. Here you're doing real world testing of real world patients with all the different species and strains that I showed you in the beginning. And because you don't have to use statistical analysis to turn down sensitivity to make it more specific and vice versa, there's no more trade-off. And so that makes it already much better. And this is why recombinant testing has really replaced the standard testing. And what are the ones? Again, it's the Lyme screen assay. That's a simple, very sensitive, broadly sensitive first-tier test. So if you have a requirement for a two-tier test, this LSA replaces tier one. Immunoblots offer the maximal sensitivity and specificity. I'll show you some data on that. Will detect and name many of the species that you're looking for. It will separately report the IgM and IgG and really pretty much replaces the Western blot. And the combination of the LSA and the immunoblots are what is now FDA cleared as a FDA approved or FDA cleared Lyme test. They have what's called a broad coverage assay, which is a more of a cost-effective, simplified immunoblot. Use the same recombinant technology, but they combine it into one little simple test, and the response is either yes or a no. So it combines IgM and IgG. It doesn't separately report them. It doesn't tell you what species it is. So simply yes or no, and basically for that reason has replaced the ELISA. The AccuDot is a home test I mentioned where you can order these online without a prescription, and that's a home version of the broad coverage assay. It's not exactly the same, but it's pretty much similar technology. I want to switch now. We'll talk about individual pathogens, starting with Borrelia, both Lyme and relapsing fever. Now, Lyme testing. Why is it controversial? Why is it that you get a test from one of those big commercial labs or a hospital system and you have a Lyme patient, you get a false negative? Why is that? Well, actually, you have to go back to the history of Lyme in our country to find out why. So in the 1970s, there was the outbreak of neurocognitive symptoms in kids and adults. Finally, the CDC came and sent an investigator who was a rheumatologist, immunologist, and of course, he thought it was an autoimmune disease. Early in the 1980s, ticks were collected from an island off of New York, Shelter Island, and sent to Willy Bergdorfer. His lab is a tick research lab, and he discovered, in fact, a spirochete in the ticks that were collected. They named it Borrelia burgdorferi. That specific spirochete, they call it Borrelia burgdorferi strain B31, and they use that to become the standard. They call that the type strain of Lyme. So that's the standard. The government standard, the laboratory standard for Lyme disease is this one. Okay, so what about the strain B31? It's the only one that's been used in the CDC, the NIH, and the university-based research when they're studying Lyme disease. It had been the only species and strain used in all of the CDC accepted and FDA cleared tests for Lyme, and still what's used by all the big box labs and hospitals, even though from the 1980s on, we've now realized that, hey, there's more out there than this one tick strain. So that's why I say it's a bad decision. It's likely that the single strain of a single species of spirochete that came from a tick and not even a human being, never tasted human blood probably, and came from an island isolated off the coast of New York, how could that reflect the whole picture of Lyme disease all across the country? We know that Lyme Borrelia adapt to their environment, adapt to their hosts, adapt to temperatures and so forth. And so they genetically change slightly depending on their area. And so to use one specific species that came from a tick from this one little place really is not very logical. So what's the result? Research based on this, I have to say, is not the best, and likely the tests are not very good. And here, in fact, is the breakdown. I'm showing you Lyme Borrelia as well as relapsing fever Borrelia for a very good reason. I'll show you why in a moment. So look at the list on the left. These are the Lyme Borrelia that we have in our country. B31, they call that Senso stricto, the strict version of it. We also have a human strain 297 and all these others that have been found in human patients in the United States. Okay, why did I highlight this? Well, again, this is what the commercial labs use, this B31 strain. They don't use any of these others. If you have an infection with Spilmania or Californiensis, you can't even get a test for that from these big labs. But when you test from IGeneX, all of these are included. Similar story with relapsing fever. You can get a test for Hermsii. Separately, you can order a test for Miyamoto. It's not such a good test, but you can get that. But all these others have been documented in North American patients, including even Mexico. But you can't even test for them from these big labs. But again, when you test with IGeneX, all of these are included. So that's a real big deal. Now, the standard serologies are not recommended for all the reasons I told you before. That's the IFA, the ELISA, the Western blot, and the microarrays, because they're made from whole organisms that are lysed. It's a dirty system. You have to cut down the sensitivity to maintain specificity. And statistically, and I'll show you the data on this, IFA and ELISA, sensitivity 50%, half and half. It's a coin toss. Specificity in the real world is probably no better than 75, 80%. Western blot is very similar, not much better. And again, I'll show you in red, it's one species, that lab strain B31. Here's a study compiled by Ray Stricker, where he took studies that were reported in the literature and tabulated them. Sensitivity 49% for the ELISA. The one I highlighted here, instead of having a specificity that's high, they toned it down to get a higher sensitivity. And again, that's that trade-off I talked about. You want to get it more sensitive and you get less specific. And that's what happens when you have these old-fashioned tests. The multi-array tests and microwells, it's the same situation. Again, the Western blot, it's a lab strain. They lyse it. All these dirty things come out. It's not very specific, not very precise. And again, it's a single species. Look at, again, lane number 8. As a lab tech or as a clinician, you can't look at this and say, oh, I know how to interpret a Western blot. You just can't. The other thing, it's a test. It's very finicky. I talked to lab people from the NIH, and they said, you change the temperature of the bath that this sits in, or change the salt content just a little bit, and you get a different looking blot. Also, how do you rate what's positive and negative? One of them joked, well, I put it through a photocopier, and if I see a band on the other side, I'll call it positive. I mean, that's how non-specific this is. So when someone says, oh, I got a Western blot, I think it's a good test. No, it is not. Now, what about the immunoblot? This has changed everything. It replaced the Western blot. It's multiple species, not a lab strain. They found it so sensitive that in early Lyme, the validation studies detected nearly all of the CDC samples that were positive for early Lyme. There's never been a test that we've had for Lyme disease that you can use in early Lyme. Some comes in with an atypical rash or a summer flu. They tell you, well, don't do a blood test now for Lyme. It's too early. Well, for immunoblot, you can do it. In the disseminated samples they got from the CDC, when they did the validation studies, they found it was 100% sensitive and specificity was 97%. So basically, you can use the Lyme immunoblot for early, intermediate, and late Lyme. And here are the data. You found, again, early cases. His early Lyme, what CDC calls stage 1, either IgM or IgG positive, 93.3% sensitivity. There's no other Lyme test you can do in early Lyme that's 93% sensitive. When it comes down to disseminated Lyme, again, 100% sensitivity. When it comes to specificity, whether it's IgM and IgG, it's at least 97, in some cases as much as 99% specific. And I call your attention to IgM. IgM itself is 99.3% specific in this series. And people have said, IgM is not always very specific. Well, in this case, it is. All right, now why is this better? Yes, it's now recombinant antigens, not the things made from lysed organisms. But what's different is, besides doing all this work to get really pure specific proteins, they use what's like an inkjet printer to print these chemicals, these antigens, on the strip. So they're not spread out by some vague electrical current in a different bath of temperatures, salts, whatever. And they're not vague based on how much is growing in a culture. They put exactly the amount they want in exactly the spot. They know what it is. And that's what makes it so highly accurate. And it's available for Lyme, relapsing fever, Babesia, and Bartonella. And in these infections, they can detect all the major species and name most of them. And of any serological test, it's by far the highest sensitivity and specificity. Now, I want to tell you something that I discovered. People said, oh, I can get immunoblot at LabCorp. I said, oh, really? So I looked it up. Turns out immunoblot is a generic laboratory term used for any test that specifically breaks apart a sample into individual proteins. So technically, a Western blot is a form of an immunoblot. Maybe IGeneX should rename it. I don't know. But if you find out what they're doing, what these labs call an immunoblot is not. It's a Western blot, one species, lab strain, and all that. And it's only IGeneX that offers an immunoblot based on recombinant technology that's able to pick up multiple species. And not to beat a dead horse, but again, side by side, there's no comparison. The Western blot's like I said, dirty. It's one species, one strain of one species, actually. Immunoblot, all these different species, and in fact, others, and it's so clear. It's very clean. There's no comparison. Remember I said IgM? Many of us clinically see patients who have disseminated Lyme, and they have maybe a negative IgG, but a positive IgM in their serologies. Well, what is that? Most infections, IgM is gone by 4 to 6 weeks. There's this natural transition to IgG, but somehow in Lyme that doesn't happen, and that's been proven, in published studies. All right, so now when you're doing a test with one of those old-fashioned standard serologies, ELISA, Western blot, if you find a late positive IgM, the powers that be say you should ignore it. The CDC, the IDSA, NIH say ignore a late positive IgM in disseminated Lyme, chronic Lyme. And the reason why they say that is because they found the old-fashioned tests will cross-react IgM with viruses and other organisms and even autoantibodies. It does cross-react, for example, with neural tissue. So they're right. Don't rely on a positive IgM if it's an ELISA or a Western blot. However, as I showed you on that validation table, the IgM Lyme West immunoblot is 99% specific. Okay, so here in this case, you can accept and believe in IgM that's positive in any IGeneX recombinant serology, even in late and disseminated disease. And it's now FDA cleared, okay? It's called the IDART. It's going to be marketed as soon as they get insurance codes, believe it or not. That should happen soon. In fact, this was kind of revolutionary. I mean, those of us who've been around a long time know that we've been fighting the CDC since the early 90s because they wanted a two-step process that included an ELISA as tier 1, believe it or not, which already screens out half the cases. And then tier 2 was a Western blot, and they said which bands to look for. They gave us 10. It's supposed to have 5 out of 10. Turns out 7 out of those 10 are not specific to Lyme disease, so that's ridiculous. The ones that are specific, 31 and 34, they excluded for political reasons. So if you look now, the CDC had to change their website, had to change their criteria. No longer do they talk about the Western blot bans. They no longer talk about 5 out of 10. They no longer mention 31 or 34. They say if it's FDA cleared, you can use the test. So that Lyme immunoblot is so powerful that it forced not only the FDA to approve it, to clear it, but for the CDC to have to change what they've been saying now since the beginning of Lyme. It's a big deal. All right. A lot of this technical talk doesn't really matter. You're a clinician. You're not going to look up statistics. You have a patient in front of you. What do you mean? FDA clearance is really important. First of all, obviously it adds validation. No other Lyme specialty, Tick-Borne disease specialty lab has this. And it's really important to support your clinical diagnosis. We know Lyme is a clinical diagnosis, correct? But in this world of, legal medicine, you really like to have laboratory backup for your clinical impression. There are patients who don't believe you when you say you have Lyme disease. There are family members. There are other practitioners who criticize you. Oh, you diagnosed Lyme in everybody. Hey, if you say, look, I have a patient who's got a test that's FDA cleared and it's been shown to be 99% specific, that's reassuring to everybody. And God forbid someone should call you on the carpet, you have this to back you up. That's why it's not good to use a lab that's out there that doesn't publish their findings. It doesn't report, like I did today, their validation studies. And I'll show you some real world. These are case series from 3 different practices. They're just practices from Lyme-limited practitioners. 3 different ones were taken. First series, 36 patients. These all had clinical Lyme, but negative Western blots. They were seronegative. Guess what? Well, they only had one. Well, let me start from the beginning. They found 10 had Borrelia mayonnae. They also found Spilmania, Californiensis, Greenii. You see the list. These would never be picked up by a Western blot because they're only designed for that one lab species. But guess what? They did find one on the immunoblot that should have been picked up by the Western blot. It wasn't. So it shows that not only does the Western blot not pick up what's really out there, what's supposed to pick up it didn't do. So that's series number 1. Series 2, 29 patients. Again, Lyme clinically, negative Western blots. They found all these other species, 2 that the Western blot should have picked up, it did not. Interestingly, 11 of them could not be speciated. What does that mean? Well, it means it was a positive, but they couldn't say in the report which species it was. Now, I said to Dr. Shah from the lab, what is that? Why does that happen? She said the most common thing in their studies has been that it's a patient who's co-infected with more than one Borrelia. It shows up so many bands on the immunoblot that you definitely know it's positive, but because there's overlap, you really can't say which one it is. In fact, even in this series, 4 were Borrelia from Europe that were unspeciated. So here, if you see an immunoblot that's not speciated, it's still positive. Don't ignore it, okay? Third series, again, same story. This one is interesting because like the others, it showed co-infection. It actually showed it because here, antibodies to 2 different ones were shown in 2 different samples. So co-infection with different Borrelia is real, just like co-infection with different Bartonella is real. I want to talk to you about relapsing fever, and people say, why relapsing fever in a Lyme patient? Well, first of all, relapsing fever is very common. If you look at the map, good old New York State where I live, blank. Why? Well, they weren't reporting there. Well, what can I tell you? But there are at least 17 species of relapsing fever Borrelia, and I count at least 8 to 10 that have been reported in humans in North America. So it's out there, okay? And there's a lot of misinformation. Don't get stuck in the trap. First of all, thought to be rare. No, it's found all over the United States. Second, well, it's not the right tick. It's got to be a soft tick, not a hard tick like in Lyme. Well, that's also not true. Number one, soft ticks are present in the United States, especially in the southern states. That's absolutely proven. In fact, they're endemic in several cities in Texas. And hard ticks have been proven to transmit tick-borne relapsing fever. So that's not true. Also, it's said to have a very distinct clinical presentation. You could not miss it, and it could never confuse with Lyme. Well, that's not true at all. It can present exactly like Lyme and therefore be not correctly diagnosed. And that's the point of this. You have a patient that looks like Lyme and acts like Lyme, gives a history like Lyme, but the test for Lyme is negative. Think about relapsing fever, okay? Now, classically, relapsing fever is supposed to start with a very high illness and a high fever and a very severe illness that lasts for a day or 2, and then bam, a big crisis where you sweat and you get lightheaded and hypotensive, and it's a crisis. But then after a period of time, you start to feel better and you feel like you've recovered over the next few days. But 5 to 7 days later, it recurs again. And that's why it got the name relapsing fever. That's a textbook definition. And probably that's not as common as thought in relapsing fever. Probably a Lyme-like presentation is more common. Case series. IGeneX looked at their serum samples, not from a dozen patients, but from 543 patients. 32, and these are patients who had suspected Lyme, referred to IGeneX from Lyme-aware clinicians. 32% indeed had antibodies to Lyme, 22% had antibodies to relapsing fever only and not to Lyme, and 7% had both. How about this? A published report. 90 patients who met the published clinical case definition of chronic Lyme disease, not suspected to have relapsing fever. Here of the 90, 46 had Lyme positive on their testing. 56, more than half, had relapsing fever antibodies, and 8 actually had both. So this is published. So don't ignore relapsing fever. Another thing, it's a nasty illness. Transmissions has been reported quickly in one series within 15 seconds of the tick bite if it's a soft tick. It's known to be maternally fetally passed. That's well recognized and accepted. Spontaneous abortion, premature birth, neonatal death have all been reported, as has pericarditis and pericardial effusion. The louse-borne relapsing fever, not from ticks, but from lice, that's been shown to be transmitted by mucous membranes. And what's crazy about that is I talked to Willy Bergdorfer years ago, and he told me, it's not published, but he told me that he thinks Lyme can do that too, go through mucous membranes. ARDS, acute respiratory distress syndrome, has been described and published by the CDC from Borrelia hermsii, relapsing fever Borrelia. Relapsing fever Borrelia can inhibit the immune system by evading complement, just like Lyme. And this is another thing that's very important. Some of these infections can prolong the QT interval on the cardiogram. And those of you who know cardiograms, a prolonged QT interval puts a patient at risk for serious and even life-threatening ventricular arrhythmias. And that's why some medications which prolong the QT interval are things that have to be looked at. So if you have a patient with relapsing fever, my advice as a clinician is do a cardiogram at baseline. If the QT interval is on the borderline side, and you start to treat them, because many medications and even some botanicals can affect the QT and make it longer, I recommend that you repeat the cardiogram once they're on treatment, just to be sure you're not putting them at risk. And it's also great documentation for your chart from a medical legal point of view. So like I said, if you're seronegative for Lyme, as I showed you in that series, maybe it's relapsing fever, because none of the Lyme tests out there, whether there be an ELISA, an immunoblot, a PCR T-cell test, none of them for Lyme have been validated for any relapsing fever species. And as I showed you, when you're looking for relapsing fever, only relapsing fever testing from IGeneX will do all the different species of importance here in North America. Okay. Now, babesiosis. As I showed you earlier, it's apparent, according to the lab data, the most common co-infection in Lyme patients, the parasite, not a bacterium, transmitted by the same tick that can transmit Lyme, and a lot of the symptoms that are non-specific to malaise or fatigue overlaps. But the thing is, when Lyme patients are concurrently infected with Babesia, the Lyme symptoms can be more severe, the immune system is more suppressed, and Lyme treatments therefore need to be more aggressive and given for a longer duration. So again, think in the flip side, if you have a Lyme patient who's really, really sick and not responding well to treatment, think Babesia. They might be co-infected with Babesia. Test them for that. Same story. You can get lab testing from the big commercial labs and the big hospitals for Babesia. What do they test you for? They test you for microti, which is only a small percentage. Very few test for Duncani, which is more common. None of the big labs test for MO1, Oticoli, or all the others on the list. IGeneX, again, will test for all of these when you order Babesia tests. Now, what's out there? A stained blood smear done in the hospital, because acutely the infection rate is very high. The rate infection by the percentage of red cells infected, and usually 3, 5, sometimes higher percentage. But that falls off rapidly, even without treatment. So within the first week is when it's sensitive. Literature says 2 weeks, but in my experience with a lot of Babesia patients acutely ill, after about the first week, it's really not such a good test. I don't believe it's good after 1 week, forget 2 weeks. IFA testing for Babesia, the standard old technology, it's outdated, it's insensitive, and the only the only Babesia test offered in many labs is an IFA, and it's for microti. Immunoblot, like I said, for others, it's more sensitive, it's more specific, it will detect all those different species. And actually, in New York State, it's the only test you can order that detects Duncani as well as the others. The FISH test is great for Babesia. It uses a fluorescent labeled DNA probe that targets the RNA of the organism, much better than a standard dye. It's genus level, meaning it can detect multiple species. And their culture is a great test. It's genus level, can detect multiple species, and they've actually found some really atypical species that no one ever expected. So that's a test to do when you really don't know what's going on. Immunoblot, very sensitive, very specific. And look, I want to call your attention to this. Here, of their sample, 63 was positive. Of those, 25 were microti, 20 were Duncani. So that means of the 63, 18 were not Duncani or microti. So it shows you that of 63, 18 out of 63 were some of those other species. So go back to slides. Something else on this list, probably Oticoliae, MO1, we don't know. But the thing is, if you don't use an immunoblot, you're going to miss things. No cross-reactivity. Here they looked against all the other major tick-borne pathogens, like the different Borrelia, different Lyme Borrelia, different relapsing fever Borrelia, Bartonella, even E. coli, no crossover. Second series were Anaplasma, Ehrlichia, Rickettsia, Lyme, and even normal controls, 100% specificity, no cross-reactivity, okay? Here's a picture. It's a fluorescent dye. They use a black light in the microscope. It's pretty neat. Now, when you do a standard smear, like I said, hospitals do for acute infections, what they call the limit of detection is how many red cells can they detect that's infected. If 3% of red cells were infected, they say it's a 3%. 1%, it's 1 out of 100. They can go down probably as low as a 0.5. In other words, 1 out of 200 cells they can pick up. If the infection is lower than that in terms of organisms in the peripheral blood, they just don't see it. They miss it. But in Babesia fish, this is published. The limit of detection is not 0.5, it's 0.001. It's hundreds of times more sensitive. It's 1 out of 1,000, not 1 out of 200. The other thing, like I mentioned, fish testing can detect these even if they're in biofilms. It's a genus level test. It indicates an active infection if it's positive. And the reason for that is this is a test looking at the RNA of the organism. RNA has a very short half-life in the bloodstream. It's a matter of hours. So if you get a positive test, when the test was done, the person was infected. Now, the thing is that if someone's already on treatment for Babesia, that may suppress Babesia counts. It makes the test less sensitive. Lyme antibiotics, especially the cell wall drugs, don't do that. So that you can use. Then you could use the test. But if someone's been on Babesia treatment and you do the test, you're probably not going to pick it up unless they've been off treatment for a period of time. So that's a good pearl to know. So when you're testing for Babesia, they're very hard to detect. No one test is 100% sensitive. And as I mentioned, they're finding some really strange atypical species that no one ever expected. So what people are recommending to do is you do a combination of tests using different methods to sort of broaden your scope. So you always start with the immunoblot in the fish. In difficult cases, the culture is added, again, to find some very atypical organisms. Bartonella, another big player in the tick-borne disease group of patients. It's extremely common, can be confused with Lyme. There are at least 45 species, more every day are being found. At least 15 of these have been documented to infect humans, and co-infections of different species of Bartonella have also been published. And why is it so common? Well, you can get it so many different ways, so many different vectors, mosquitoes, even biting flies, mites, red ants. I got Bartonella from being attacked by deer flies. Of course, cat scratch, dog bites, that kind of thing. We know that ticks now can transmit it. It's been proven finally. Maternal fetal, of course, needle sticks for veterinarians. It's worldwide distribution, as you can see from the map, even above the Arctic Circle. So it is a real big deal. The Bartonella now is, their immunoblot is now available nationwide. This is a validation study comparing it to other tick-borne diseases. There is no cross-reaction with Lyme, relapsing fever, Babesia, Anaplasma, individually or as a group. And then when they found positives by other tests, be it FISH, IFA, or Western blot, if you have positives in that, you're going to pick it up pretty much all the time on the immunoblot. So it's a really good test. Bartonella can be tested by FISH also, again, biofilm testing. Bartonella is a big time biofilm maker, including in the bloodstream, so it's great that the test will pick it up. Genus level detection, so you are able to pick up multiple species. They've been valid to detect this whole list you see here and others even possibly. And these are the pictures. I love pictures. See the fluorescence, you can see the organisms, little chain of dots here. Here's a negative control, of course, is negative. The FISH performance, here specificity study was 100% specific, whether it was testing against Babesia, different Babesias, Borrelia, Plasmodium even, trypanosomes, Anaplasma, Rickettsias, no cross-reactivity, which is really important. And inclusivity study, these are patients who had these other species confirmed by DNA sequencing after positive PCRs, including one they didn't know what the species was an unknown species. It picked up even that, 100% inclusivity on these samples. So again, it's a very broadly sensitive test. Now, fish testing, I want to talk to you about that. This comes back to the days, again, in the 1980s when I worked with Alan McDonald when he was doing this type of testing with Lyme. Um, for a fish to be called positive, you have to have three things. Number one, you have to see the fluorescence, obviously. Number two, you have to find the specific morphology. In Babesia, you have either the ring form or what's called the Maltese cross. In Bartonella, you can see the organism, Coccobacilli, and here you see it there. And the third thing, the location either in the cells like in Babesia or associated with the cells with Bartonella. Sometimes the stain can precipitate out and give you what they call in the lab stain dots, and these little dots here and there. And that should not be called a positive because it's not the morphology, it's not associated with the cell, and it's not certainly not uniform in size like you see here and you'd expect. I know Alan McDonald was very heavily criticized. They thought, again, he was overcalling Lyme disease. The powers to be didn't believe him, but he went to great lengths to prove that, in fact, these things were stain dots and these things were real. So when you do a fish test, insist that you have a lab like IGeneX who will not call this positive. Okay, there you go by the three rules. You can get a T cell assay for Bartonella. Again, it detects specific T cell responses very early. Someone comes to you, they had an exposure, let's say a few days ago, they finally get into your office and say, wow, I'm all swollen. It looks like I have cellulitis. The T cell test is sensitive enough early enough so you can do that. Also, late in the disease, when antibody levels get low, the T cell balance goes up. Seronegative patients doesn't mean you're going to be T cell negative, it could be positive. Another thing I like to mention, if you can do an immunoblot, which reflects B cell reactivity, and also do a T cell response assay, the IGeneX spot, that's a T cell response. You're getting a B and a T cell. So if you know someone has a disease, in this case, say Bartonella, and they're responding with the T cells, but not the B cells, that's a good indication that there's something wrong with their immune system. They're not responding. And that can give a clue to do further studies and see what's really going on. And like with the other T cell assays that IGeneX offers, it's extended species coverage. Recommendation here, just like Babesia, very hard to detect. They consider Bartonella to be a stealth organism. So what you need to do, because it's stealth, multiple species, you need to do whatever you can to try and diagnose this from a lab point of view. So you always start with an immunoblot. People always recommend you add the fish. You may have to add on the culture or the T cell assay, the IGeneX spot, or all four of them, because you really need to pin this down. Sometimes it doesn't show up. You may have to repeat the test. It's been reported that for this, as well as with Lyme disease, it's not been published, but it's been reported by clinicians that after treatment, sometimes the sensitivity increases, and there are reasons for that. Also, we know that Lyme and Babesia are immunosuppressive. So if you start to treat Lyme and Babesia, that's also present, and/or you can repeat the test for Bartonella after that, because now the immune inhibition from the Lyme and Babesia is lessened since it's under treatment, and now you can start to pick up Bartonella being positive. And this is very commonly seen where Bartonella test was negative in the beginning, you treat the Lyme, and wow, and now the Bartonella is coming positive. Clinically, another interesting thing happens. Someone presents to you like Lyme disease, and you have the swollen joints and the neurocognitive and the stiff neck, the whole thing, and you treat them for that. And little by little, as the Lyme gets better, they start showing up with Bartonella symptoms. It's like it uncovers a suppressed infection, and now clinically you know what's going on, and then the Bartonella testing is actually more sensitive. For early disease, the FISH test, like I said, is a very great test early on because it's a direct smear as well as a T cell assay. Rickettsias, we're going to end on this. The testing that's offered at this point is an IFA. I know an immunoblot is in the works, but right now it's an IFA, but you can get a culture. It's a big advance over the standard PCR. It will detect many species, including some atypicals they've picked up. And that includes Ehrlichia anaplasma and the basic rickettsias, but not Rocky Mountain spotted fever. The government doesn't allow culturing of Rocky Mountain spotted fever unless you're biosafety level 4, but the standard rickettsia culture has accidentally picked up some Rocky Mountain. So basically, because rickettsia can be very severe if it's acute and can linger chronically and cause low white cell counts and so forth, unexplained liver enzyme elevations, you really want to test with both methods if you can. So as you can hear from what I've been telling you, IGeneX tests are more sensitive, they're more specific, they're more inclusive of a lot of different species than any other commercially available ones. IGeneX will freely tell you their validation results. They show you the FDA clearance of the Lyme immunoblots. The immunoblots for Lyme even have a package insert. You can get that online because it's FDA cleared. Now, this is important for validation, important for peace of mind, and for your security as a clinician. As I showed you, it's vital to use test methods that include many different species. Don't rely on the outdated tests. And for that reason, I recommend, and so does the lab, that you do a test panel. And they offer test panels at IGeneX that combine complementary different methods on multiple species to increase the yield and also to save costs. The panels are less expensive, apparently, than if you do them a la carte. And just so you know, the most commonly ordered tests in 2025 at IGeneX were the immunoblots and the Babesia. I'm sorry, the immunoblots and the FISH for Lyme, Babesia, and Bartonella, and all the others. The most prescribed test panels, they call them TBD panels, TBD 4, 5, and 6. My preference, what I like the most is 5 because it includes immunoblots and FISH. If you want to add on PCR, you can do the 6 as well. So just as FYI, people say, what panel? It's a very confusing test directory. What do I do? Well, just do the 5 IBL and you're covered. And if you're not sure, which again, it's a complicated test menu, I recommend you reach out to IGeneX. You can call them directly. You can definitely talk to the sales staff, your sales rep. They can help you with test ordering, give you clues on getting better insurance reimbursement, how to interpret the results. Sometimes they can get me involved if they really need someone who's a clinician who can give you some backup. And don't forget the website. There are discussions of the test methodologies, there are webinars, research papers, sales materials that make it very easy to figure out, and pricing, of course. And they have an 800 number.